The enzyme-substrate complex is formed when you have your enzyme, represented by the color yellow, and your substrate, which is represented by the color blue, coming together. When these two join together, this is the formation of the enzyme-substrate complex. The enzyme changes its physical form as the substrate bonds. The enzyme allows it to speed up the chemical reactions in the body, allowing it to join two reactants together or break down a reactant into two or more products as shown in here. The substrate can only join with the enzyme through its active site, which is made to perfectly fit the substrate. One example of this is through the enzyme protease, where it binds with the protein to break it down into smaller amino acids through hydrolysis.
Competitive inhibition occurs when an inhibitor and a substrate compete with each other to bind to the active site first. In these situations, the inhibitor, in this case represented by the color dark blue, binds to the active site before the substrate, represented by the color light blue, blocking the substrate from binding with the active site and inhibiting, also known as reducing or stopping, enzyme activity. A real example of this is when malonic acid and succinic acid compete for the active site of succinic dehydrogenase, in which the malonate, the inhibitor, beats succinate, the substrate, to the active site, inhibiting enzyme activity and therefore slowing down the Krebs cycle and cellular respiration.
Non-competitive inhibition happens when an inhibitor binds to a different part of the enzyme, not the active site. This part is also called the allosteric site. When the inhibitor, represented by dark blue, attaches to the allosteric site, it changes the enzyme's shape. Because of that, the substrate, represented by light blue, can no longer fit into the active site, and it ceases the entire reaction. A real-life example of this would be when lead or mercury enters the body. These heavy metals can bind to enzymes in your cells and change their shapes, preventing normal reactions like the ones that help make energy from happening. Allosteric regulation happens when another molecule attaches to a different spot on the enzyme called the allosteric site to control how the enzyme works. If an activator shown in dark blue binds there, it helps the enzyme work faster by making the active site fit better with the substrate. A real-life example is how oxygen affects the enzyme hemoglobin in your blood. When one oxygen molecule binds, it helps the enzyme change shape so that more oxygen can attach easily. This is known as positive allosteric regulation.
Feedback inhibition is an enzymatic process used by the body in order to regulate the amount of products coming from a certain enzyme. This process is used to maintain homeostasis within the body. With feedback inhibition, the substrate enters the enzyme and gets broken down into the products. However, instead of, in this case, both of the products leaving away from the enzyme, one product is used as an inhibitor to stop the reaction that the enzyme is doing. This is shown here as we see the active site of the enzyme becoming closed. One real-life example of this is through the creation of ATP, as when a cell has enough ATP, one of those ATP molecules will bind onto an enzyme in the glycolysis pathway, resulting in the blockage and preventing overproduction of ATP.
The denaturation of an enzyme occurs when the enzyme's active site loses its physical structure, causing the substrate to be unable to bind to it. Every enzyme has an optimal pH and temperature range, and when the pH or temperature falls out of that range, the enzyme will denature, in other words, become deformed, and the reaction cannot take place. A real-life example of this is when salivary amylase, the enzyme in saliva that breaks down carbohydrates, denatures. This occurs due to body temperature rising above the optimal temperature of 98.6 degrees Fahrenheit or 37 degrees Celsius, causing denaturation due to high unoptimal temperature. When salivary amylase, which has a neutral pH, interacts with stomach acids, which have a pH of 1 to 2, it also causes denaturation due to abnormal, unoptimal pH.